Myself, Dr. B.S. Londhe, working as assistant professor in chemistry, Karmavir Bhaurav Patil Mahavidyalaya, Pandharpur. And this is the first part of the chapter Pharmaceuticals. Pharmaceutical chapter for this syllabus. It has following points. First of all, we will see introduction, then qualities of an ideal drug, methods of classification of drugs, and method based on therapeutical action. This is for our syllabus. Brief idea of penicillin G, only constitution, that is the structure, synthesis and uses of the following drugs. In this, antimalarial, anti-TB, CNS, anti-diabetic, anti-inflammatory, antibiotic, and anti-cancer drugs, there are for our syllabus. Now, let us see some introductory points. The term pharmaceutical is used for any chemical that find use in the preparation of medicament, that is medicine for internal and external administration of human body. Pharmaceutical chemistry is the chemistry of substances used in medicine. So it is the chemistry of substances used in medicine. It makes use of general laws of chemistry to study drugs, that is their preparation, chemical nature, composition, influence on an organism, and study of physical and chemical properties of drugs. The method of quality control and the conditions of their uses. In other words, it is the chemistry of drug. So, what is pharmaceutical chemistry? It is the chemistry of drugs. The word drug is derived from French word drogue, which means dry herb. Drug may be defined as a substance used in preparation, diagnosis, and treatment or cure of the disease in human or other animals. According to WHO, that is World Health Organization, a drug is a substance or product which is used or intended to be used for modifying or exploring physiological systems or pathological states for the benefit of the recipient. The compound possessing therapeutical value, that is curative value, are known as drugs. Due to administration of drug, body overcomes ills and maintain normalcy. According to Paracelsus, life is essentially a chemical process and greater proportion of changes that takes place in body are chemical. So, and hence ills of the body must be treated by chemicals. Paracelsus told that for the first time that life is a chemical process and ills of the body can be treated with the help of chemicals. So, drugs may be obtained from different sources such as plant. From plant, we get morphine, quinine, reserpine, etc. Animals, from animals, insulin, thyroid, heparin, minerals, mineral sources. are of mercury, gold, copper, arsenic, antimony. From microorganisms, we get penicillin, bacitracin, and synthetic sources, aspirin, procaine. From genetic engineering, we may obtain hormones. So these are some sources and some examples of the drugs obtained from sources. The earliest reference about medicine preparation in writing comes from India in Rig Veda and from China in their Materica Medica, 2500 to 3000 BC. In India, later on, a large number of medicine preparations, including Ayurveda, were described by physicians Charak, Shushruth, Vagbhat, and others. All these drugs were of vegetable or plant origin. Western medicinal system comes from Egypt in their literature that is known as papyrus. Hippocrates, 450 BC before Christ, laid down the foundation of modern medicine. According to him, a disease is a pathological process and its treatment with drugs is not a magic. For the first time in history, Hippocrates stated that disease is a pathological process and its treatment with drug is not a magic. The medicine system introduced by Hippocrates had scientific basis. It was based on observation, analysis, and deduction. However, up to 19th century, it was believed that treatment of disease was mainly based on combination of guesswork and experience. So Hippocrates laid down the foundation of modern medicines. In general, disease or diseases are caused by one or more of the following types of germs or microbes, such as bacteria, protozoa, viruses, fungi, worms, etc. Because parasitic diseases, the difference between these germs is in their size. So, protozoa, the diseases caused by protozoa are malaria, dysentery, syphilis, sleeping sickness. From bacteria, leprosy, tuberculosis, cholera, these are the diseases caused by bacteria. And viruses cause polio, smallpox, mumps, HIV, that is human immunodeficiency virus. Paul Ehrlich, 1909, the father of modern chemotherapy. Remember, Paul Ehrlich is the father of modern chemotherapy, has introduced the term chemotherapy. Chemotherapy means the treatment of disease due to bacterial infection by by chemical compound. Treatment of the disease by chemical compound. That is known as chemotherapy, which destroy the microorganism without affecting to any material extent the tissues of the host. In 1907, Paul Ehrlich used the methylene blue in the treatment of malaria. The following steps were employed in testing of medicinal value of the chemical substance, and testing can be done in vitro or in vivo. So, in vitro testing, The microorganisms which cause a particular disease are cultivated in the culture. The chemicals and the taste were applied and growth rate of the microorganism was studied. If the compound kills or retards the growth of the microorganism, then next step is carried out. That means in vitro testing is the testing which carried outside the living body in test tube, in petri dish, etc. In vivo testing, it is the testing inside the living body. So, in vivo testing, the microorganisms which cause a particular disease are injected to the animals such as mice, rabbit, monkey, guinea pig, etc. And disease is allowed to develop. After the development of disease, the chemical under taste is introduced orally or by means of injection and effect is studied. If it retards the further growth of or kills the microorganisms, then it is subjected to direct testing on patients in hospital. That means in vivo testing is the testing inside the living body and in vitro outside the living body. Depending upon the action of drugs, the chemotherapeutic agents are divided into two groups, namely bacteriostatic agents. These bacteriostatic agents only stop the growth of bacteria or retards the growth of bacteria. And bactericidal agents, these chemical compounds or drugs, they kill bacteria directly. The drugs which kill the microorganism are on bactericidal. So bacteriostatic only stop the growth and bactericidal directly kill the microorganisms. Static and cidal effect depend on several factors such as concentration, pH, temperature, duration of the action, metabolic phase, presence of interfering substance. Thus, drug with static effect may exert cidal effect if the dose is increased. Thank you.